Hello and welcome to Groups 4 Pig's Kidney Dissection video. Today, we will be exploring the structure and function of the pig's kidney, and we will be identifying its parts and tracing how blood is filtered and turned into urine.
Now, we'll begin dissecting the kidney. Using a scalpel, we'll make a gentle cut lengthwise, starting from the outer curve and moving toward the hilum. Careful to not to cut all the way through, so that we can open the kidney like a book and observe the structures inside.
If we look closely at the inner curve of the kidney, we can see the small indentation called the renal hilum. This area serves as the entry and exit point for three important structures that are the renal artery, the renal vein, and the ureter connecting the kidney to the rest of the body.
At the hilum, we can identify the ureter, the renal artery, and the renal vein. The ureter, which is often surrounded by extra fat tissue, extends downward from the kidney and serves as the passage that carries urine to the urinary bladder.
Once the kidney is open, we can see three major regions inside. The outermost layer is the renal cortex, which appears lighter in color.
The renal cortex that contains millions of nephrons, which are the functional units of the kidney. These nephrons are responsible for filtering the blood. Within each nephron are the glomerulus and the Bowman's capsule, where the filtration process begins.
To follow is the renal medulla, which contain the cone-shaped renal pyramids that look striped because they are filled with collecting dust. Between these pyramids, we can also find the renal columns, which hold the blood vessel that connect to the cortex to the renal artery and renal vein. And at the tip of each pyramid is the renal papilla, where urine drains into smaller chambers called the minor calyces, which joins to form major calyces before converging into the renal pelvis.
Lastly, the renal pelvis is located in the middle of the kidney. Urine is collected by this structure from the collecting ducts. Urine then travels to the bladder via the ureter and is held there until it is expelled from the body through a process known as maturation.
Now, let's trace the pathway of blood through the kidney and the movement of filtrate that eventually becomes urine.
Blood first enters the kidney through the renal artery. This artery branches into smaller arteries and arterioles until it reaches the afferent arteriole of each nephron. From there, blood enters a cluster of capillaries called the glomerulus, where filtration takes place inside the Bowman's capsule.
Once filtration occurs, the filtered fluid, now called filtrate, flows through the renal tubules. It passes through the proximal convoluted tubule, goes down the loop of Henle in the medulla, and continues into the distal convoluted tubule. As the filtrate travels through these tubules, the kidney reabsorbs useful substances such as water, glucose, and ions. This process ensures that the body retains what it needs while removing waste and toxins.
The remaining fluid, which is now urine, flows into the collecting ducts, then down through the renal pyramids and into the renal pelvis. From there, it exits the kidney through the ureter and is stored in the urinary bladder until exhaustion.
It's incredible to consider the kidney as a sophisticated recycling system that filters blood, stores essentials, and consistently keeps the body in balance.
Through this dissection, we have observed how each structure of the kidney supports its function, from the nephrons in the cortex that filters blood to the pelvis, that collects urine for excretion,
Highlighting how intricate and efficient our bodies are when it comes to maintaining homeostasis, regulating fluids, and eliminating waste.
That concludes our pig's kidneys dissection activity. Thank you for watching, and remember to stay hydrated and exploit your hardworking kidneys.